You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Richard Jacobs with the Future Tech and Finding Genius Podcast Network. I have Evan Jones. He's the CEO of OpGen, a publicly traded company, OpGen Incorporated. And we're going to be talking about their work. So, Evan, thanks for coming.
Thank you. It's great to catch up this afternoon.
Yeah, tell me, what's the premise of OpGen itself, and then we'll talk about some of the particular projects that are in play right now.
Sure. Well, OpGen's a genomics and informatics company that is focused on improving the patient management of patients with acute infections, in particular antimicrobial resistant infections.
Yeah, I know that a lot of bacteria are developing antibiotic resistance. So, yeah, what's the premise on how you would attack such a thing? Is it with a new drug, or is it a new way to attack bacteria in general? I mean, what's the overall thought process there?
So the field has been, come a long way since antibiotics were discovered in in World War II. Yeah. And so over the last 60, 70 years, there have been diagnostics that have come to market to help improve patient management. But typically, it's taking 2 to 4 days to get the right diagnostic information to make a precision medicine guided clinical therapy decision, for instance, to get the patient on the right antibiotic. With the new genomic technologies that we have at our disposal, you can run a DNA test in just a couple of hours. If you have the informatics in place to interpret those results, within 2 to 3 hours, you could give a critically ill patient diagnostic information that previously could take 2 to 3 days.
A lot of people don't have 2 to 3 days, and they may die before that time, or something else may go into a coma, or who knows. So yeah, that's great that it's gotten a lot faster. Are you looking at the patient's DNA, or you're looking at, again, all the potential particular offending bacteria, if it is a bacteria or virus?
So typically what we're doing, and our lead product, for instance, is for urinary tract infections. And so you'll take a sample of urine and test the bacteria that are present in that patient's urine to look for what are the pathogens that are present, what's their level, is it high enough to be clinically significant, and then to look for antibiotic resistance genes, and if they are there, to be able to predict, what's the right way to treat this patient.
So is it more about identifying the particular pathogen, or is it pathogen plus, is it antibiotic resistant?
It's both, really. I mean, particularly as an example in UTI, they want to be sure that you actually have a urinary tract infection. And that's a big clinical issue that, frankly, we've been focused on. If you look at that field, there are about 10 million urinary tract infections a year. About 8 million of them are in situations that aren't really complicated. They may get an antibiotic prophylactically, or they'll run a test and wait a couple of days and get the results. If you are in an emergency room, or a critically ill patient, and they think you have urosepsis, which can lead to a broader sepsis for the patient, you don't have 2 or 3 days to wait. So what's happening today is oftentimes they'll make a judgment call. They'll say, I think there's a pathogen there. Just in case, I'm going to give them an antibiotic. And that used to work, but with the rise of multi-drug resistant infections, about 20 to 30% of the time, if you used the routine antibiotic, you would be undertreating the patient. And by this, the inverse of that, if you said, okay, I'm worried about the 20 to 30% of patients that are resistant, so I'll use the stronger antibiotic, then you over-treating 70 to 80% of the patients.
What about if you have antibiotic resistant or multi antibiotic resistant bacteria, and you're treated with a standard or even a strong antibiotic? Does that further worsen your ability to fight that bug, or is there no harm in in undertreating?
The over-treatment and overuse of antibiotics has been really one of the driving factors behind the rise of antimicrobial resistant pathogens. It's very important to get the right drug to the right patient and the right information to the clinician. If they don't, patients can worsen. I'm sure you or some of your loved ones know people that have been in the hospital with acute respiratory infections or some sepsis, and they're waiting days for results. And of course, all the while, the patient's getting sicker. Yeah. Or, we did some research at one of the prominent hospitals in Boston, and they said, when they're faced with one of those situations, they'll give the patient up to 5 different anti-infectives, which can include, an antifungal, an antibiotic, maybe even some antivirals, just literally nuking the patient. And it's very expensive, but all these drugs also have side effects, so it's not good.
Yeah, no, that makes sense. So I guess the worst case is you do this testing, you find out quickly that, there are no known antibiotics to treat the patient, but at least in many cases, you'll be able to treat to the appropriate level.
Yeah, you hear a lot on the TV and in the media about superbugs, and they are really rising at an extraordinary rate. But most of those pathogens are treatable by some antibiotic. The trick is to figure out which one. And of course, it doesn't do any good if the patient's already died or the infection's gotten to a point where it's hard to turn around. But with these rapid tests, I think you have a better shot at that. Our products are still going through the FDA, so I don't want to give you the sense that everything is, ready to go and on market. But our FDA clearance for our lead product, we're working to get sometime in the first half of this year. We're just finishing answering all of the questions from the FDA for our 510K submission.
Once this testing is in place and it's commonplace, a lot more data will be gathered too. A lot more clinical data on what's out there. Instead of just being able to say, "Oh, superbugs are on the rise." I guess if the data was aggregated, we'd be able to say, "Oh, MRSA, this particular strain is on the rise, and this one seems to be decreasing, and this one this and this and that." So I guess we'd be much better informed as to the real picture of what's happening.
Yeah, we're involved in some of that work right now in real time with a digital health initiative that is taking place in New York State. And there are 3 large health systems involved, and they are using our technology to track in real time the pathogens that are either, in patients that are in the hospital or patients being admitted. And then the next phase of this project will be to use that information and try to improve patient outcomes by trying to drive down the rates of some of those pathogens, get faster treatment for patients, things like that.
So we spoke about it before, but if you don't mind recapping, so what is being tested now? Is it the person's DNA plus bacterial and bacterial DNA? Is it RNA? Is it proteins? I mean, what's being looked at? What can you say?
Yeah, well, it's a technically more sophisticated question than just on the surface, because if you take a sample from low respiratory for a patient or even their urine, there will be a mix of human DNA and pathogen DNA. Our tests look at the pathogen DNA. That's what we're focused on.
Okay. And then in looking at the pathogen DNA, are you looking at the the particular genes that they express, or the genes that they contain, or metabolites they make, or proteins they express? I mean, what are some of the specifics?
Yeah. So what we do, for instance, in our AMR gene panel for urinary tract infections, we have probes that are designed to specifically detect the pathogen. So they use a technology called polymerase chain reaction that amplifies the DNA of the microbe, very specific regions that are specific to each pathogen. If they're there, then the bug amplifies, and you can detect it using this what's called PCR technology. It's quite robust and proven, and very sensitive. The threshold that is indicative of clinical infection varies across patients, but it can be as low as 1,000 bacteria. And generally, our test is correlating with the number of bacteria on a one-to-one basis, so that we've picked DNAs that we believe, if the DNA were to measure 100, that would mean there were 100 microbes there.
And I guess what could be done now with the speed of this is that you could sample a patient, now, and then you could sample them, an hour later or 6 hours later, and you could see progression of certain bacteria if that's what was happening. If you could see things ramping up quickly and in a shorter time window.
You can, but in practice, the healthcare system, there's a lot of inertia there, and what's happening is that you have that patient. Let's just go through a typical patient management example of a acutely ill patient that's in an emergency room. If they believe that they have a urinary tract infection, and it's a significant infection, not just one of the 8 million a year that you could wait a couple of days. So in those acute cases, they may start an antibiotic as soon as they see them. And with our technology, they would run the test. They would get the answer back in 2 or 3 hours, so the patient could still be sitting in the ER. And then on the next dose, they would either move them over to an IV antibiotic, if it was a superbug and multi-drug resistant pathogen, they would use a stronger antibiotic than normal. And then they would continue to monitor that patient over the next several days. And the way clinicians are thinking about it currently is those confirmatory tests would come over the next 48 hours.
Right, it's better to act sooner than wait.
Yeah, but hopefully if they've got them on the right drug, by 2 days, they're starting to improve.
Right, that's true. Is there any need to go faster? 2 to 3 hours is great. Is there any need to try to do it within minutes?
Well, it would be great if we could get this technology so that it works at the bedside for any number of cases. But I think we're a few years away from that.
How much is this in use today? What's the rollout look like, or are you not there yet?
So in the case of the OpGen test, we've been in front of the FDA for the last 18 months. As I mentioned, we're just at the finish line. We've done some large clinical studies showing how the test works. Some of that was published last spring at Geisinger Health and Intermountain Healthcare, two very large health systems, showing that with this technology, they could detect over 90% of the pathogens. And similarly, about 90% of the time they got the antibiotic decision correct. So, the path in the US is that we've got to get through FDA before it can become widely adopted in the clinical environment.
Well, what's the current accuracy ratio for identifying the right pathogen? You said you can go up to 90%, but what's it against the background though?
Well, it's always an interesting question in the diagnostic field, because you have these reference methods. For instance, in the case of microbes, you have culture as the reference, and you grow the bug in the presence of antibiotic. And in theory, that method is 99%, 95% accurate. But that's not really the case in actual practice, because you can have the bugs that don't grow. For instance, in the case of a respiratory infection, the guidelines indicate that up to 50% of respiratory samples have unculturable pathogens. So it's always interesting to compare, and our test is going to give different adjunctive information to be used in conjunction with existing methods.
No, that's great. Is it how long again before it's available in many hospitals and in widespread use? What's the path?
Yeah, so it can take 6 months to a year after FDA clearance to get these technologies onboarded into hospitals. In the case of our AMR gene panel, if we receive clearance in towards the back half of the first quarter, you could see the test being used certainly in the second half of the year in major health systems. As I mentioned, we're already being used in these pilot cases, and the UTI test, we are in clinical trials currently for our FDA registration, and the goal is to submit over the summer and ideally going into this time next year, or I should say it a different way, at the end of 2020, hopefully we'll be near getting that FDA clearance for our UTI test.
And what other medical needs? I mean, so UTI seems to be very important. Any other medical needs you've identified that are first on your list to go after?
So one of the really attractive areas is for lower respiratory infections, patients that have pneumonia and acute pneumonia. And OpGen is just now in the throes of combining with a company called Curitis that today received, or it was announced today that they received on Friday FDA clearance for a lower respiratory test that in 4.5 hours can help determine the pathogen and provide antibiotic resistance genes. So that's a real breakthrough, and there's a lot of excitement about that.
Yeah, that's great. Okay. Other initiatives? I mean, this is probably funny enough, but other initiatives that you're working on?
So, the work in this field is arduous and expensive. So getting this, what we call the AMR gene panel for 47 antibiotic resistance genes has been the primary focus of our team at the company. One of the exciting parts about that is using the data, meaning the data from the DNA test, to predict antibiotic resistance. And we have a technology platform that we call Lighthouse that takes the DNA tests and matches them against a database of microbes where we've done the DNA testing and we know their antibiotic resistance data by classical methods. So that's what was used, for instance, in the UTI studies. You take the DNA test, you interrogate this Lighthouse knowledge base, and that can give you the information to help guide the patient management. And that same technology we're using in this tracking of superbugs project, where we create a profile for the bug that you could think of it being like a zip code. And it's been very encouraging in the early days of that project. The people that are running the tests and the laboratory folks, they check first thing in the morning to see if there are matches among the pathogens, and then to try to track down and figure out what does it mean, what's the new information.
Well, very good. So what's the best way for people to find out more and to keep tabs on your progress?
Yeah, so, like many companies, we have OpGen.com as our website. We also have a active social media program where we tweet. I believe it's under OpGen. We try to get a lot of news, both related to OpGen, but also related to the field in general. So those are two good ways.
And then the last question, what I don't know if you can quantify numbers, but what could be the impact of your work on people's health in terms of ballpark numbers?
Well, I think it's going to be very impactful. One of the reasons that I got into this field was seeing these data about the rise of superbug pathogens and the predictions that by 2050, more people will die from drug-resistant infections than cancer or some other large diseases. And what's exciting about these products that we're working on is I think we can bend the curve here and improve patient outcomes and allow for significant advances and improvement in treatment of patients with acute infections. Just examples that I've given you today of urinary tract infections, or the recently cleared Curitis test for lower respiratory tract infections. And I'm also confident, even though it's been very difficult to progress, I think this tracking of superbugs in the healthcare settings is going to create a mentality where we know what's going on, and people are taking steps to try to drive down the rates. And it could be that several years from now, people look at this and say, hey, we've made a big difference. The projections that said, you'd have those rates in 2050, that we beat it. It was the new technology made a difference.
That's great. That's very good. Well, Evan, thank you for coming on, and I appreciate it. And you're doing good work that's going to really benefit a lot of people. So thank you very much.
Thank you, and thanks for getting the message out.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.